wait a minute. Okay, could somebody confirm you can hear me?
Yeah.
Great, thank you. Um, just to be clear, I don't remember what I mentioned in the lecture, but I have graded to some people, about half the people now, the worksheet to lab 3. And my hope is to finish that tonight. Maybe during this lab, I'll finish it. It looks like I'm done over half the people. Yeah, I'm about 2/3 of the people are graded. Um, so I hope to have this finished tonight, and then you can submit it again by this Saturday for the second grading. For lab 4, I'm not sure when I'm going to have the first grading done. I'm not going to have it anytime soon. So I've moved back the second grading due date until February 10th. I hope to be able to start on that maybe tomorrow. Uh, but the worksheet for lab 4, the first grading has not gotten done, and I'm not sure when I will get started on it. Even though if I get started on it on Wednesday, I certainly won't finish until Friday. I've got that many of them, and they will get longer to grade. Any questions about the grading? All right. Let's move on then to lab. Let's see. Let me start sharing my screen again. Nope, that's not what I want. There we go. The syllabus. Today is January 30th, and we should be working on Lab 5. You need to also read the unknown project instruction file on Canvas for next week. Next week, I will be talking about the unknown project, and you will be starting the unknown project next week. Let me show you where those files are. So come to the main Canvas webpage, scroll down to week 5 lab. Lost my mouse. The unknown instructions.rtf is the document I want you to read. I will talk about it next week, but I would like you to at least pre-read it, look through it, so you have some idea about the unknown project. Everything else you can hold off on until I've talked about them. I will talk about the sample journal. I wonder why that one isn't indented as far as the others. The unknown list of the unknowns, building a dichotomous game plan, and then the grading for the unknown report. So I'll talk about all of those. But I would like you to look over the unknown instructions. Any question about any of that? All right. So let's talk about the lab. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. There is some reading to do, as in all labs. Not too many pages this time. Read about culture media, and then read about obtaining pure cultures. In today's lab, there will be some supplemental material. We will be watching some video clips. There is no virtual lab. The learning objectives for this lab: 1, understand the importance of proper specimen collection and transport to the microbiology laboratory. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate the various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. 5, define the following terms: selective media, differential media, and reducing media. Any questions about what we're going to cover? All right. The ability to isolate pathogens from a clinical specimen submitted for culture in the microbiology lab is only as good as the collection techniques and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, they'll dry out, and some are sensitive to the exposure to oxygen. Most anaerobes are. And these organisms, as well as really all organisms, require special transport medium to keep the specimen alive. Once the specimen is received by the lab for microbial culture, microbiologists employ the 5 principal I steps to process it. There's first inoculation, 2 isolation, 3 incubation, 4 inspection, and 5 identification. Inoculation is the placing of the specimen on an artificial culture medium to initiate growth. Isolation is the spatially separating out microbial growth into distinct colonies. Usually, especially when you have a clinical specimen, you're going to want to look at distinct colonies and maybe even pass them through 2 colony formation events so that you can grow up pure cultures. 3, incubation. That's the placing of the culture medium in an environment with conditions to facilitate the growth of the microbes. 4, inspection. After growing the culture, you then examine it for the presence of microbial growth and make a few rough characteristics and determinations as to what it might be. Not what it is, but narrow down what its possibilities could be. 5, lastly, identification, performing specific tests to ID any pathogenic microbe that's been isolated. The 5 steps will be described in detail, and we will also discuss the different types of culture media. In talking about the different types of culture media, you are already familiar with general purpose media, such as Na, nutrient agar, Na, nutrient agar with yeast extract, and TSA, tryptic soy agar. These allow you to grow many organisms, but not everything will grow on general purpose media. Fastidious organisms will not. To promote the growth of fastidious microbes that have very specific nutritional requirements, we need to grow them on an enriched general purpose media. For example, blood agar contains 5% sheep's blood added to the agar to help organisms grow. One fastidious organism that will grow on blood agar, but will not grow on a general purpose media, is Streptococcus pyogenes, the microbe which causes strep throat and a whole slew of other diseases. But the point is that you need to add additives to the media to get Streptococcus pyogenes to grow. And a simple way of adding these additives is to add red blood cells, and then the red blood cells supply the nutritional requirements for Streptococcus pyogenes. Another fastidious organism that I just read about within the last week is Treponema. I used to know this, Treponema pallidum, the organism that causes syphilis. People have been trying to get this bacteria to grow in tissue culture for over 100 years, and they never discovered what it was that they needed to add to the tissue culture, the media, to get syphilis to grow. Well, in the last couple of years, apparently they finally figured out what it is they need to add to get Treponema pallidum to grow. And so only in the last couple of years have we been able to grow syphilis in the tissue culture. Any questions about any of that? All right. So there's general purpose media, there's enriched general purpose media, and then there is selective media. Selective media is a media that contains chemicals that inhibit or halt the growth of some organisms. So it selects for some things to grow and selects against other things. Keep in mind two things when using selective media. First, a selective media isn't necessarily perfectly selective, meaning if given enough time, an organism which is selected against can grow on the plate. Second, even those organisms that are supposed to grow on the selective media may form smaller colonies or may grow a little slower on the selective media than they do on general purpose media. Any questions about that? All right. Besides selective media, we also have differential media. Differential media is a media that contains chemicals that produce visible differences among different groups of organisms. So if you have two different species and you grow it on a differential media, you can see differences in the bacteria or in the media. These differences are unrelated to how well the organism grows on the media. They're usually visible, visually distinguished from each other, meaning it allows visual differences, either different color or some other different characteristics that you can visually distinguish two different types of bacteria. Lastly, we have reducing media, and these media promote the growth of anaerobic bacteria because low or no oxygen conditions are required to grow these organisms. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. McConkey's agar is selective in that it contains ingredients that prevent the growth of gram positives. What those are, you don't need to know this, I'm just telling you, is that McConkey's media contains bile salts, which you can find in the intestines, and that's why gram negatives grow around bile salts. But gram positives don't like it, so they don't tend to grow. And it also contains crystal violet, a dye which gets in the gram-positive cell wall and then prevents the cells from growing. So on McConkey's agar, generally gram-negatives grow and gram-positives do not. And this one is actually pretty good at being selective, meaning usually the bacteria either grows or it does not. If it grows, you can also use it as a differential media to differentiate its ability to ferment the sugar lactose. If the bacteria can ferment the sugar lactose, it produces acids which will lower the pH, which will change the color of the plate to be pink. So this bacteria can ferment lactose, the sugar lactose, which will lower the pH, making the media look pink. And you notice the cells also look pink. If the bacteria, on the other hand, cannot ferment the sugar lactose, the cells will not look pink, and the media is not looking pink either. Another media, which I guess medium, which it can be more than one category, is blood agar. It is both an enriched general purpose media so that you can grow some fastidious organisms on it, like Streptococcus pyogenes, will grow on blood agar, will not grow on general purpose media. So it is an enriched general purpose media, and it is also a differential media. You can differentiate between the different hemolytic patterns of different bacteria species with blood agar. Hemolysis is the lysing of red blood cells, and different bacteria have different hemolytic patterns. There can be gamma hemolysis or gamma hemolytic activity, and that's shown by the bacteria in, let's make that close to white. It's a dark white, but it's a white, which is shown in the gamma here. Let me blow that up a little more. And you can see the red blood cells are red all the way up until we reach the bacteria. So there's no hemolysis of the red blood cells in these, with at least to these bacteria. No hemolysis is called gamma hemolysis. That's the Greek gamma. That's the letter G in Greek alphabet. And that's the way it actually looks. That's the lowercase gamma. There's also alpha hemolysis, where the red blood cells are red up until a dark ring around the bacteria. So the off-white color of the alpha is the bacteria, and you can see a dark ring of the blood around the bacteria. It's most noticeable and biggest inside the alpha. All of that is darker than here. And of course, this ring is darker too. It's a very thin ring, but it's a very distinct ring right there. The darkening happens when some of the red blood cells are lysed, but not all of the red blood cells are lysed. And this is called partial hemolysis or alpha hemolysis. And that will be a darkened area or ring. It can also be a greenish tint. And that right there is kind of dark, but I've seen it greenish as well. And I'm not sure why it's green. It's got something to do with the properties of light going through the blood. The third hemolytic pattern is beta hemolysis, where the red blood cells are completely lysed. In the beta, which is a white bacteria, we have it growing in this beta shape here. And around the beta and inside the beta, we have a clear zone where all of the red blood cells have been completely lysed by the beta hemolytic bacteria. You can actually hold this plate up to something and see through the plate. I don't know why, but I always like to look at the clock or the light. You can look at the light and then see the light bulb through the plate. Or I guess you could look at the clock and determine what time it is. Doesn't matter. But this region where the auger is clear, as well as this region here, you can see through it. You can't see through the beta because of the bacteria being there. And you cannot see through the blood auger because the blood will stop the light, so you can't see through it. You should note that there is a ring of alpha hemolysis around the beta hemolysis, and that is pretty normal. I've always seen the ring of alpha hemolysis around the beta hemolysis. Remember, beta hemolysis is complete hemolysis, where the red blood cells are completely lysed. Any question about any of that? One more. All right, this table is showing you the different types of media. We have different general purpose media, such as tryptic soy agar or TSA, nutrient agar or NA, or nutrient agar with yeast extract, and that's abbreviated NA. It promotes the growth of many organisms, but not fastidious organisms. We also have a differential and enriched medium, and that would be blood agar, which you can grow some fastidious organisms, and you can also differentiate between the hemolytic pattern of the bacteria. We also have a enriched general purpose media, and that's all it is, and that would be chocolate agar or CAP. I never mentioned in the last lab, the difference between blood agar and chocolate agar is that you add the blood cells to the blood agar after the agar has cooled to between 45 degrees Celsius and 50 degrees Celsius. And then when you add the blood, it's not hot enough to lyse the blood, but it is hot enough to keep the agar liquid. And then you just mix the blood in and then allow the plate, the agar to cool further, so then it'll form the blood agar. And chocolate agar is the same, except you're adding the blood as soon as you get the media out of the autoclave machine. So it's very hot. It's close to 215 degrees Celsius. And when you add the blood at that temperature, it will lyse the red blood cells and change the color of the plate from blood red to chocolate colored. So there is no chocolate, but apparently whoever invented this media thought that it looked like chocolate, and so they called it chocolate agar. You don't need to know about chocolate agar and why it's called that. I'm just informing you. It's kind of interesting. We also have selective and differential media. We have 3 of them: McConkey's, abbreviated MAC; eosine methylene blue, abbreviated EMB; and hectone agar, abbreviated HE. These are selective and differential. They have chemicals in them that will select for the growth of gram-negative cells, and they will select against gram-positives. And then they have other chemicals in them to differentiate between different types of cells. They are all differential on the basis of their ability to ferment the sugar lactose. If you ferment the sugar lactose, the plate will turn a different color than if you do not ferment the sugar lactose. The color will depend on which media we're talking about. McConkey's will turn pink if the bacteria ferments the sugar lactose. If you grow it on EMB, the bacteria will turn dark or a metallic green color if the bacteria ferments the sugar lactose. On the other hand, if you grow it on HE plates, the bacteria, if it ferments the sugar lactose, will turn a salmon color. Salmon is in between yellow and orange in color. And it will not be salmon colored if it cannot ferment the sugar lactose. We also have selective media, and by selective media, I mean selective alone, and that would be phenyl ethyl alcohol, or PEA. And this media selects for the growth of gram-positive cells. It selects against gram-negative cells. So if the cells are growing on a PEA plate, it's likely to be gram-positive because the gram-negatives are selected against. And lastly, we have selective and differential media. It's a little different than the selective and differential above. It's mannitol salt agar, or MSA. This one is selective in that only organisms that like 7% sodium chloride or higher can grow on the plate. And that's because the plate has 7.5% sodium chloride in the media of the plate. Most organisms cannot grow at over 2% sodium chloride. So most organisms will not grow on an MSA plate, mannitol salt agar. So that's why it is differential. I mean, selective. It'll grow if it can tolerate 7.5% sodium chloride. It will be selected against and not grow if it does not tolerate that high concentration of salt. And just to give you a bearing, the salt in your body is isotonic, and it's 0.9% sodium chloride. So 7.5% is close to, is that close to 10%? 10 times, folks, excuse me, 10 times more. So 7.5% will be 0.75. I don't recall. So, anyways, it's a lot of salt in the mannitol salt auger plates, and it's at least 7.5% more salt than an isotonic solution, which many organisms prefer to grow in isotonic solutions. Mannitol salt agar is also differential in that it can differentiate between a mannitol fermenting species and a non-mannitol fermenting species. If the bacteria can grow on the plate, you can assay for mannitol fermentation. But if the bacteria cannot grow on the plate, you cannot assay for mannitol fermentation. If the bacteria can ferment the sugar mannitol, that will produce acids which will change the color from a pinkish color to a yellow color. And if it cannot ferment the sugar mannitol but can grow on the plate, then it will not be yellow in color. It'll be whatever color the bacteria is. Any questions about any of that?
Can I ask a quick question about the table? I was just looking at your table that you're showing versus the table that I'm looking at that I downloaded from Canvas, and there's just a couple like little differences on the comment section. I don't know if you know if one of them is more updated. Like,
I'm not sure. What's the difference? What does yours
well,
say?
yeah, I was trying to figure that out. Um, so like for example, on the second row where it says TSA with 5% sheep blood, you have it highlighted. It says differential and enriched medium, but mine says enriched general purpose medium. And then there's a couple
All
other
right.
ones that are
An
a little.
enriched medium is the same as an enriched general purpose media. I probably made it smaller just to fit in this box.
Oh,
I've got it
okay.
highlighted because I want to make sure I specifically talk about what I have highlighted.
Right.
Okay.
Okay.
Is there any other differences?
Yeah, so let's
Go
see. The
on.
last one that says mannitol salt,
Uh-huh.
yours says selective and differential, and mine just says selective.
Oh, really? That's a mistake.
Oh no, sorry. You know what? It says selective, and then the next sentence actually says also differential.
Okay,
So
that's the
it's
same.
probably fine. Sorry.
Yeah, that's the same.
Yeah.
All right. Like I said, I've got it highlighted just to make sure I cover those areas. All right, inoculation is just putting the cells you want to grow on a plate or in a tube. Isolation was started by Louis Pasteur, who came up with the concept of a pure culture. Generally, when you run a test, you want to run it on a pure culture because if you were to run it on a mixed culture, one containing different species in it, if the test were to become positive, you wouldn't know which species in the mixed species would be the one that made the test positive. So normally in microbiology, we always work with pure cultures. Louis Pasteur did come up with a way of obtaining a pure culture, and that is he got a liquid, grew things in liquid culture, and then took dilutions of his growth, and he would take many different dilutions, and then he would replica plate to each dilution 10 times. And if in one of the dilutions, he had only one tube with growth in it and 9 tubes with no growth in it, then he correctly assumed that the one tube with growth in it was most of the time a pure culture. It wasn't always a pure culture, but most of the time it was a pure culture. And that's because probably one cell got in the tube and then grew, creating the growth that was seen by Louis Pasteur in the tube. Robert Koch developed a much easier way to get a pure culture, and that was he came up with streak plate method, where you streak cells on a plate to get separate and isolated colonies. And then you take the cells from one colony and streak it out again for isolated colonies. And if the second time you get all of the colonies the same, then you can assume that you have a pure culture. Now note that to get a pure culture, Robert Koch had to streak out the cells at least 2 times. And that's because if you streak it out 1 time, you will get the majority of the cells to be pure, but not all of the cells, the colonies, will not be pure. And in fact, 30% of the colonies will be formed by more than 1 cell. And if a colony is formed by 2 cells, 3 cells, or a clump of cells, that will not be a pure culture. Any question about any of that? And so you streak it out 1 time and you'll get 70% of the colonies that are formed from pure culture. So 70% of the colonies will give rise to a pure culture, but 30% will not. And 30% will be from a mixed culture. And 30% is just way too high in microbiology. And that's why we say you need to streak from the cells from a single isolated colony on the first plate and streak it out for colony isolation on the second plate. And then you can pick the cells from one colony and either use that as your stock culture all term long, or you can just use it for testing for the like the feet. I have some students who wore the costume we have in microbiology, or maybe the mascot of the Clark College athletes. And the point is that to get a pure culture, you need to streak it out 2 times from a colony, picking the cells up from the first isolated colony and then streaking it out for isolated colonies on the second plate, and then pick the cells from one isolated colony on the second plate to set up your stock culture. And you will be working with a pure culture 95% of the time or higher. And that's good enough to use in microbiology and call it a pure culture, even though less than 5% are pure, but some are still not pure. By pure, I mean only one species is in the tube. All right, theoretically, the last quadrant should have individual bacterial cells that will be plated and then go on to grow isolated colonies after incubation. However, it doesn't have to be the last quadrant. It can be an earlier quadrant. It just depends on how dense the bacteria are when you first start streaking them. I'm not seeing the picture now. Did I go past it? I'm not sure where I have the picture. All right. You then have incubation, and that's just putting the culture media in an environment to facilitate the growth of the microbe. We in the Clark College Microbiology Lab have fairly simple incubators, and all they do is heat, bringing the microbes to either 30 degrees Celsius or 35 degrees Celsius. We have incubators for both temperatures. That's why we use two different temperatures. And our incubator also obviously can control the time. You just stick it in at a certain hour and then pull it out another time. Most students like growing things for 48 hours so that if they set it up in the first lab, like on Monday, they could harvest it on the second lab on Wednesday. And so that's another thing you consider. How long you grow the cells is a thing about incubation. And then our incubators can also humidify the environment. What you do is you just put water in a dish, and there's the dish containing the water, and then shut the door. That will cause the water to evaporate, and that will humidify the air in the incubator. Like I said, this is pretty important because if you don't add the water to increase the humidity, then when the incubator is on, it'll warm up the media and it'll pull water out of the media. And that will eventually dry up the media. And then your cells will die unless they can form an endospore. So incubators heat, they always heat, and they usually also humidify the air. By humidifying the air, you can keep your culture along longer than if you did not humidify the air. Any question about any of that? All right. This incubator is showing you a much fancier incubator. Besides controlling the time, meaning you stick it in and then when you stick it out, the plate. It also controls the temperature. It does control the humidity. And then on top of that, it controls the amount of oxygen the cells will be exposed to, and it can control the amount of carbon dioxide that the cells can be exposed to. Some cells prefer less oxygen than normal air, room temperature air, or standard temperature and atmospheric pressure, I think it's that, stamp. I've got one more thing to talk about. What the heck was it? And then other cells prefer carbon dioxide to be a little higher than it is in our normal air. And the only one I know off the top of my head is VD prefers to grow in a reduced oxygen environment. It does require oxygen to grow, but it actually grows better and faster when oxygen is not elevated to the normal oxygen level. And this is probably just something that syphilis has evolved to because it's growing in our body tissues. And especially when it's done damage in that tissue, the oxygen levels will decrease. And That's why this venereal disease prefers to grow at lower oxygen temperature percentage than whatever the oxygen percent of this plate was. I don't remember. Well, normally the plate will just grow out in the air, and so the oxygen in the air can move into the dish and then move into the agar. And so, at least at the top, you'll be growing your microbe at full oxygen full oxygen exposure. But it doesn't have to be that way. Like I said, you can grow it in an anaerobic condition, or you can have it in a fancy incubator and then lower the oxygen and raise the carbon dioxide, or vice versa. I don't know of any microbe that grows better than current atmospheric oxygen, but I bet there are some. I just don't know them off the top of my head. 4 is inspection. You then observe the visible colony morphologies between the different species of bacteria. You can even begin narrowing down what your possible species are by looking at things like the colony size, the colony shape, the colony margin, and the colony elevation. One species will have the same for most of these, and that's why we can do this with inspection. You then do identification, and that is run specific microbiological tests to try and identify the species of bacteria. Once you begin the unknown project, you'll understand this procedure for identifying the species in much better detail. Any question about any of that? All right, know the terms of this lab. You can skip the references. You're not going to be doing any of this activity in real life. Everything will be done online. But in this lab, we have a little video on inoculating an agar plate using the streak plate culture technique. And in this video, you only need to watch to 4 minutes and 15 seconds into the video. After that, you do not need to watch the movie any further. We have a little clip on beta hemolysis, a second clip on alpha hemolysis, and a third clip on gamma hemolysis. So go ahead and read those. There is at least one question where you will need to find the answer by getting information either in this lab module or from somebody else who just happens to know. And then we have a little video on McConkey's agar. Once again, you only need to watch that video into the video for 3 minutes and 50 seconds. After that time span, you can cut out the material and it'll be fine for the plate. And then lastly, we have a video on mannitol salt agar. Go ahead and watch that. We do not have a virtual lab for this lab. And then answer the questions. Let me go to the worksheet for lab 5. 1, describe a natural circumstance where staphylococcal species of bacteria might benefit from salt tolerance. So when in the natural world would E. staphylococcus benefit from being in a natural environment containing salt so that the Staphylococcus can grow and other species cannot. So you need to name a natural circumstance. You can't say like growing in a mannitol salt agar plate, because that's not a natural circumstance. 2, how might hemolysis benefit a hemolytic organism? So we have a bacteria that can produce hemolysins. That's something that causes hemolysis. And then how will that bacteria benefit from the blood cell being lysed? Three, imagine you have a culture containing a single bacteria species. This culture could be one of many possibilities. So one of many different species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify your known bacterial species. That is, how would you use these plates to help you identify the species? Now, I'm going to warn you that each of these plates has 2 properties that we can use to help you identify the species. So you must describe 2 different species, 2 different characteristics of the plate, the media, to get full credit for this question. If you only give the one property from each plate, you'll only get half credit on this question. Any question about any of that? All right. Question number 4, what's the main goal of streaking for isolation? Meaning streaking for colony isolation. What's the main goal of it? 5, what happens to the growth on the plate if you forget to sterilize the loop in between sectors when streaking for colony isolation? You need to be specific, discussing the growth you see on each sector. So let's go back to the lab module. Go out to the streaking. Why am I not seeing the streaking? Let me see if it's in the previous lab. I'm just trying to get a plate showing you colony streaking. Yep, that's the lecture. I don't want the lecture. I don't think it's in here. I wonder if I did something by mistake and got that fouled up now. No, that's not well. I'm on lab 5. That's the picture I'm looking for. Why couldn't I find that in the other? See, for some reason, it's supposed to be here and it's not. I'm not sure what happened to it. Let's come back here. And let me copy it in here. Hopefully I can grab that entire table. All right. So when you streak, you normally sterilize your loop at the beginning and then pick up your cells from your stock culture and then streak out sector 1. You then take your loop and put it in the bacterial incinerator to get it red hot and then cool it and then do streak 2 and then sterilize your loop and do streak 3 or sector 3. Well, in this case, you're doing everything the same, except once you streak out quadrant 1, you do not put the loop into the bacterial incinerator and then bring your loop over here to streak out sector 2, where you're getting the cells from sector 1. If you do not put your loop inside the bacterial incinerator between sectors 1 and 2, you will have you'll have less dilution of your cells than you would if you do the streaking and then you put your cells into a dilution. So normally what we have is like 1-to-1, and that's probably discussed. And then you have 1-to-10, which what you do is you take 1 mil out of the 1-to-1 and add it to 9 mils. And then that makes the 1 in 10 dilution. Anyways, on this question, you do the quadrant streaking. However, you don't take the loop after quadrant 1 and sterilize it to streak out sector 2. And the same with sector 3. You don't, between sector 2 and sector 3, you don't sterilize the loop. You just continue streaking. So you have cells both from what is on the loop as well as the cells you're picking up from sector 2 on the plate. And then you do the same thing between sector 4 and 3. Any questions about any of that? Actually, I don't want that. Let's go to the worksheet. So problem 5 is a thinking question. Students often get question 5 and 6 wrong. So you really do need to think about the answer before you write it up and then turn it in. 6, what would happen to the growth on the plate if you transfer from stock culture to inoculate each sector? Be specific, discussing what you see in each sector. So let me demonstrate once again. So normally you sterilize the loop, cool it, pick up cells from your stock culture, streak out section 1. Then normally you cool the loop, heat up the loop in the back to incinerator, and then cool the loop. And then you streak out sector 2. And where you get the cells from sector 2 is you're getting them from sector 1. And so you should be streaking at least lightly or slightly into sector 1. But you do not want to drive your car all the way, because you'll just pick up a whole bunch of dust and it'll get on your car. And then you do this for each of the other sectors. So in this case, you start sector 3 and you pick the cells once again for your stock culture to streak out sector 3. And then you do the same for sector 4. When you're about to streak out sector 4, you put the loop in the back to incinerator, cool the loop, put it into your stock culture, pull out the cells, and then streak them on the plate of media on my desk. Any question about any of that? All right, remember 5 and 6 are difficult. You need to think about it before you give your answer. And it's probably a good idea that once you give your answer, think about it, go through and read the question and read your answer, and make sure that the question is the correct question to answer the questions. All right, 7. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That means initially you were working with a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? And be specific in your answer. 8. In the table below are a variety of agar media that are available to microbiologists to cultivate bacteria in the lab. We don't have this many media, but we could, and probably at one time in the past, we had all of these media. If not, like maybe, where is it? Phenyl ethyl alcohol. Maybe this term they do not have phenyl ethyl alcohol, but they'll probably add a antigen containing cells here, and hopefully the immune system won't react to it and then kill it off. Anyways, on this table, mark an X if you think the media belongs to one of these 3 columns. Like nutrient agar, if you believe it's selective, mark an X there. For let's go on to blood agar. If you think it is non-selective, then make an X there. I'm trying to think, this is a little trickier because we don't have normal leather soles on your shoes, but that'll be good enough. And then with BAP, the blood agar, mark if you think it's selective and differential. With chocolate agar cap, do the same. Mark where you think it belongs. And then do the same for all of these. You can find the information to this table, to fill in this table, in the lab module. All of them are mentioned. Any question about any of that? All right, the last question, number 9. One of these plates was inoculated using streak plate technique. State which plate it is. Is it plate A, plate B, or plate C? All right, any question about this lab? If there's no questions, I will be here till 8:30 to help you with the lab and any questions. However, once the last student is logged off, I will log off as well. If there's no questions, I'm going to stop the recording and I just stopped the share.